Hello, everybody. Good afternoon. Seems like we're online. I've been having a bit of weirdness in the back end, but looks okay now. I probably shouldn't talk about weirdness in the back end on a Thursday afternoon. Right. Okay. Let's make a start. So we're up to number 36 now. That number's ticking ever upwards, I guess, is the nature of numbers quite often. Yesterday we looked at annotations. So we spent quite a bit of time with Biomart, both the Biomart package in R, the bioconductor package, and also we had a look at the website as well because it was a reasonable kind of comparison piece to look at looking up data and looking up annotation on the Biomart website and how that compares to the usage of the R package, because they are very related. Because really, what the R package is doing is absolutely looking up data on the Biomart website. So that's what we spent yesterday doing, and then at the very end, we redrew our interactive volcano plot and added in the gene names, the external gene names that we had acquired from Biomart. And then when we hovered over the genes in the volcano plot, we not only got an idea of what the fold change was and what the p-value was, but also what genes they were, what genes it was that they were referencing. So we got the actual human readable gene names for our for the points on our volcano plot, and that was extremely that makes that volcano plot much more useful than it did than it was with the ensemble gene IDs on. The ensemble gene IDs are only are not very human readable. They're very useful, as we found yesterday, for mapping to other data sets, but they're not massively useful for genes which we want to be able to, if we want to be able to see what the genes are. And so that's where we're at. We've got our volcano plot. So this is our interactive volcano plot here, and we get an idea of what the genes are on that volcano plot by hovering over the points, and we get the gene names popping up in our little viewer. Although admittedly this doesn't look like it's being very responsive to me at the moment. But this viewer, I've found, despite the fact that my MacBook is really not a bad machine at all, it really struggles with this interactive plot with 15,000, 16,000 points on it, which I guess is not massively a surprise, but nevertheless is a bit of a pain. And so for machines with fewer resources than I have here, this might be even more of an issue. I'm not sure if it would work better if it was popped out in its own little browser, as opposed to sitting inside RStudio. So if you click in the open in a new window little pop out button here in RStudio, it will open it in your browser. So it's opened it up for me here in Chrome, which I've now managed to lose. I don't know where that's gone. Oh, there it is. Okay. Whether that is more performant, whether the browser deals with it better, I'm not really sure. It doesn't look like it's massively better. I'm just trying to get it in a browser where you can see it. Bear with me 1 second. There we go. So there you go. So this is there. That's now popped up in Chrome rather than in RStudio. I'll be honest, it doesn't look like it's going to go very much faster in Chrome versus RStudio. I think it's just there's an awful lot of data that sits underneath this plot, which makes it slightly difficult to work with. That's a little bit better, I guess. So you've got SAA1 and SAA2 are two of the most robust p-values, as well as CXCL8 and CCL20. Some of these are familiar genes to me from S100A8. These are genes which have cropped up in other studies that I've worked on. Got this one all the way out here, which looks like an unannotated gene. This looks like something we might not necessarily want to work with. But anyway, so these are this is our interactive volcano plot, which actually is not too bad in the browser. It's better. It looks like it's better in the browser than it is in RStudio, a little bit at least. Okay, so that's that. Let's shift that out of the way, and let's remove it from here as well, just to make RStudio work a bit faster. So what we're going to do today then is to take a look at a third visualization of these kind of results. We've looked at MA plots, and now reasonably extensively volcano plots, and we kind of used having that volcano plot as a motivation to go and get hold of that data from Bioinformatics, which will be useful to us in other purposes as well. What I want to do specifically today is to draw a heat map for the first time. So heat maps are an extremely popular visualization of gene expression data. There's lots and lots of ways of drawing heat maps, and I want to show you a couple, and so that we can talk a little bit about the pitfalls of heat maps and how we could possibly go about doing it better. What I'm going to do is I'm going to do a heat map of our differentially expressed genes. So our 141, I think it is, differentially expressed genes. And we're going to look at the expression of those differentially expressed genes across our 6 samples that we're working with. So we're going to have, let me get my whiteboard, which I don't have open at all anywhere. So I'm well prepared today. As with yesterday, I was in a meeting until 5 minutes ago. So this is just working from home life at the moment. I suspect it's probably the same for lots of people. There's lots of meetings, lots of different video conferencing software. I think I experienced my 8th or 9th different video conferencing software yesterday, which is one I'd not come across before. And consequently, lots of an awful lot of kind of sitting in front of a camera, trying to remember not to pick your nose. Right, so that's whiteboard loaded on here. Just need the same on so that you can see it, and then maybe I can draw what I want to draw. I haven't been able to pin Jacob down today for any length of time. He's been very all over the place, so I'm afraid I don't have a picture of the day for you. I am trying, but it's not always possible, and he seems to be doing a lot of schoolwork at the moment. Right. Okay, this is taking a long time. Slick as always, Simon. I'm sure if I was any slicker, you would all abandon me in droves, because you would wonder who I was and who had abducted me, which aliens had abducted me. Right, I'm going to stick this on the screen so you can see it spinning away as well, just so you can see that I'm not just filling time for no reason. So the basic idea behind a heat map, anyway, is effectively what you're doing is you're representing a matrix of data in a visual way. So rather than looking at a table of numbers, what you want to do is you want to show that table of numbers in a visual way. So the way that you would normally think about a heat map is having rows and columns exactly the same way as a matrix does. So we're going to have, I'm going to get this wrong, 1, 2, 3, 4, 5, 6 columns, because we've got 6 samples, and then we're going to have 141 rows, which I'm not going to draw. And so then the heat map colours these the cells in the heat map according to the expression of that gene in that particular sample. So we're going to have 141. I'm not going to do red and green, but we would have all of these cells, all different colours, depending on the expression of that gene in that particular sample. And then the other thing that distinguishes a heat map from a tile plot or something like that, where it would literally just be a representation of your data, a direct representation of your data. Sometimes they're called quilt plots or tile plots, where you're just taking your matrix and putting it up on screen. Is that a heat map also has clustering applied over the top of it. So we will end up with a dendrogram linking the samples together, but also the genes as well. So we'll have different clusters of genes drawn out in our heat map in various ways. So yeah, that's the thing that kind of distinguishes the heat map from just having a visual representation of a matrix, is that you're able to apply this clustering, and the functions that produce the heat maps in R are capable of also producing that clustering as well. And so we'll be doing, it's kind of doing 3 things simultaneously. So it's not just a straightforward visualization. Okay, so there are lots and lots and lots of ways of producing heat maps in R. There is a base heat map function called heatmap, which will take a matrix of data and will draw a heat map from it. We're going to do that to begin with, and then talk about the reasons why that heat map is not ideal. Okay, and then we'll look at some alternative ways of doing it. There are lots of ways of making heat maps that are variously complicated, and they're complicated in lots of different ways. So for the purposes of today, I'm just going to pick one that isn't. So I'm going to pick one that isn't the base heat map. But there are lots of others, and we can talk about some other ones that are available as well. They're good for different heat map packages are good for different things. But the first thing that we need is a matrix of data that we're going to plug in to our heat map. And that data should really take the form of, it's going to be a matrix, and we need the counts for our differentially expressed genes. And we can do it as the normalized counts. But the scales of normalized counts can be kind of all over the place. So maybe it might be better to use the VST transformed counts for a heat map, because they're probably in a better scale and will work better for what we're trying to achieve here. So I think that's probably what I'm going to do, is I'm going to get the appropriate values out of our VST transformed counts for our heat map. So the first thing that we need is the identifiers for our differentially expressed genes. So if you remember, our differentially expressed genes ended up, and this is a couple of days ago now, so we need to go back a bit, in this, the final results of our filtering earlier in the week, in filter_df3. So if we go back and remember the dimensions of that, 141 genes is what we've got. Actually, what I'm going to do, the first thing I'm going to do is I'm going to reapply the filtering, but I'm going to do it on our new annotated data frame so that we can pull the annotation forward as well. So if we go back to our filtering up here and just copy out the relevant lines, so that is this one here and this one here. Let's copy those and take them back down to the bottom of our script. Okay, so our annotated data frame is called annotated DF. So we're going to filter that in the same way as we filter DF1 before. So we're going to filter on the basis of the adjusted p-value, and we're going to call this anno DF2 just to distinguish it from the previous one. And then we're going to filter that data frame on the basis of the absolute fold change. And that will get us to anno DF3, which will still have the 141 rows that filter DF3 had, but we'll have more columns. So we'll have 15 columns, and those columns include one of those columns is the external gene name, which will be useful for labeling up our heat map when we get that far. So in there are our the ensemble gene IDs for our differentially expressed genes. They're in the first column, which is the ENS gene column. So our differentially expressed genes, or DEGs in this case, are the identifiers which are found in anno DF3 and the ENSG gene column. So we're going to pull that out as a vector like that. So that's our ENS gene column as a vector. So there's 141 identifiers there. Now we can use that to pull out the rows from the dataset which contains the expression data that we want. And we already have that data. It's the variant stabilization transformation data that we generated way back when we did our PCA. Although actually we may not, maybe we don't have it for this. So let's create it. So our DSeq dataset is, so we want VST NHBE is the results of the variance stabilizing transformation for our DSeq dataset for NHBE. Okay, so this is going to give me the VST transformed normalized counts for our 6 NHBE samples, which are the samples that we have been analysing so far. And then the actual data that's contained within this. So this VST NHBE thing that we've just made, variable that we've just made, is a data set of type DSeq transform. So it's not a matrix, it's not a data frame or anything like that. It's this more complicated data set. And we actually want to extract the VST counts from that as our data set. And so I'm going to call it the VST and HBE matrix is in the assay slot of the VST and HBE object. So this will extract a matrix of the VST transformed counts. So again, we can actually, let's look at it like, so VST NHBE mat, and let's do the first 3 columns, the first 6 rows. Done that before several times, just to show you those numbers there. So that's our sorry, first 3 rows, first 6 columns. That's That's the counts for those 6 samples, all 6 samples in those first 3 genes. But what we actually want, so this has the dimensions of this is large, because this is all of our, not just the 16,000 that we analysed for differential expression, but all of the genes that we inputted into DE-seq in the first place, which is some 60,000 genes. And we don't want to heat map all of that. If we put that matrix into heatmap, it would take the rest of the day to generate the figure, but also it would be completely illegible, and wouldn't be any use to us at all. So what we actually want to do is we want to cut this down to just the differentially expressed genes. So this matrix has row names. So the row names are the ensemble gene IDs. So we can filter it by using row names. So if you want to filter on the basis of the row name, you can put ensemble gene IDs into, let's get a couple, just by way of example. So that's the first one. This is the third one. Okay, and then we want to filter on the rows, so we put that selection before the comma, and then we don't filter the columns, so we don't put anything after the comma, and that will give me those 2 rows back out. So if we want to take a selection of the rows in this matrix, we can use the ensemble gene IDs for the relevant rows to do it, and that's what we have in this DEGs vector that we created a couple of lines back. That is a vector of the ensemble gene IDs which we're interested in, because they're our differentially expressed genes. So in order to get our data for our heat map, so I'm going to call it data for HM, we want to filter VST NHBE mat using our differentially expressed genes. Okay, so that will pull out just the rows for the data which corresponds to our differentially expressed genes, the 141 rows. So the dimensions of this, if we go back to using dim again to find the dimensions of that matrix, you can now see that has 141 rows and 6 columns. So that is the data which we want to visualize using a heat map. And we can generate a heat map from that data without any kind of configuration. We don't need to do anything else to it. We just run the heatmap function with data for HM as an argument. And this will generate a heat map. This will give us our first heat map straight away. So there you go. That is that data in heat map form. And base heat map is, I mean, it's all right. It's not. There are a number of reasons why it's not fabulous. So we can see the differential expression in here. We can see that these genes are differentially expressed, but we can't tell anything about the magnitude of the expression in any of these rows because we have no key. And the base heatmap function gives you no way of generating a key to tell you what's what here. You can configure like the size of the row names and the column names in order to get them to fit on the screen properly and things like that. But there's a lot of configuration that's missing here. This data is also scaled by row, which actually for this kind of data is fine. So you can see that you can always see which side is low and which side is high. Although actually, because I don't have a key, I'm not actually sure which side is low and which side is high. I think that the reds are the cooler colours and the yellows and whites are the hotter colours. So these will be higher numbers and these will be lower numbers. But without a key, I can't be sure about that. And there's lots of reasons why this isn't the best way necessarily of drawing a heat map. It's certainly the fastest way, but I'm not sure that it's the most effective way. And so there is quite the market in packages for R which are capable of producing better heat maps than this one. So there's a package called gplots, which has a function called heatmap.to, which is comes with things like a key automatically and various other ways of configuring the heat map to make it slightly better than this one. Then there are packages built specifically just for producing heat maps, and some really good ones as well. So pheatmap is one of those, and complex heat map is another one, which really does, as the name suggests, lets you build really very, very complicated heat maps. Today, we're going to use pheatmap because I think it's a good balance between producing nice looking heat maps by default and configurability. So being able to work with those default quite nice looking heat maps to turn them into even nicer looking ones, and heat maps that look the way you want them to look. Just as a quick side note, one of the things you might be thinking is, well, why don't we do this in ggplot2? You know, this is a visualization, and I've talked about using ggplot2 for visualizations and how flexible it is and how it's able to produce really nice looking figures. I'm not convinced it's the best. There might be a way to produce a heat map from ggplot2. I'm not upfront aware of it. The problem isn't getting your kind of visualizing your matrix, because there's a geometry called tile, which lets you color tiles in on your ggplot, so you can produce the kind of inner part of the heat map in ggplot perfectly fine. It's attaching the dendrogram to that and reordering the rows and the columns on the basis of the dendrogram that I'm not even sure how to go about doing that in ggplot, and I've never really explored it in any detail. So consequently, we use other packages for this. So we're going to use pheatmap to improve on this heat map that we've drawn here. So first of all, we need to install it. So I'm not sure if I've restarted my R session or not. So let's Biop Manager : So if you're ever not sure if you've attached a package, especially if it's a like Biop Manager, which we use for installing packages, which it's not going to be part of the library calls in your script. If you want to be really explicit about this is the function that I want to use, you can tell in which package the function is located. And so that's what I'm doing here. I'm saying from the package Biop Manager, run the function install, and it's the two colons that allow you to kind of join those two things up. So package : function is being very explicit about the function that I want to run. Okay, so then the package I want to install is called pheatmap. I've heard people call it pheatmap before, but I don't really like that. So pheatmap is what I call it. So this Biop Manager will now go away and install pheatmap and any dependencies that it has. So there we go. I'm still not updating those packages. It's very persistent. So let's stick it in up here on our libraries. So library pheatmap. Load the package. And again, we're just going to, we'll run this just with the defaults so we can see the difference between the base heat map and the pheatmap. And then we'll try and do some configuration. We'll fiddle about with it a little bit. Okay, so here we go. Right then, this is the pheatmap, so the equivalent to pheatmap of the matrix that we pulled out of our VST counts. Now, this looks very different to the equivalent p-heatmap, the equivalent base heat map. And there are quite a number of reasons for that. The first and most obvious difference is the colour scheme. So the default colour scheme for p-heatmap is very different from the default colour scheme for the base heatmap function. So that goes from red for the low counts to white for the high counts. And in pheatmap, we go from, you can see on the right, we do actually have a key, which is one of the other big differences, but it goes from blue for the low counts to red for the high counts through white in the middle. So that there is one of the big differences, is this difference in color scheme. The other big difference is the base heat map. So if we go back to the base heat map, this is what's happened here with this heat map is they've applied scaling by row. So the colors in each row are between, they work out the z-score of the expression in that row. So they, in order to scale by row, you subtract the mean. So you take the observation, you subtract the mean, and you divide by the standard deviation, and that gives you this z-score for across the row. So it means that the counts are scaled within the row. So you can always see within each row which set of samples is showing a high expression and which set of samples is showing low expression because of that scaling. But that also means that I can't compare the expression of the gene in the first row with the gene in the last row, because they've been scaled differently, because they've been scaled according to their own counts. By default, in pheatmap, you don't get that scaling. It isn't scaled by row. So by default, what's being plotted here is the actual values in your expression matrix, so in our VST matrix. That makes it slightly harder to see on a per row basis, to see which is the sample which has the high level of expression and which are the samples which have the low level of expression. You can figure it out, but it's slightly harder to figure out. And it's a slightly more honest representation of the data. So you're actually seeing the true magnitude of difference in the actual count data here. So there's that, and also you can compare between rows. So you can look across this data and see whether one gene is expressed to a high level or a low level relative to the other genes in the dataset, as well as within the actual between the actual columns themselves, between the samples in the experiment. The sort of side effect of that for this data is that actually this isn't the best colour scheme in the world. Having a diverging colour scale, which this is, where you have an inflection point around the middle and you go from one colour, one type of colour to another type of colour from the bottom of the scale to the top of the scale, is not actually ideal for this kind of very sequential data. So there's nothing about the middle point here, this sort of 10 on this scale, where this inflection happens. There's nothing about that number which means that you need to completely change the color of your point of your cells in your heat map. It's an arbitrary thing, and actually a continuous color scale would probably be better for this data. So shades of red or shades of orange or shades of blue or something would probably be a more appropriate choice for this data. And so this is an interesting representation of this data. I still don't think it's perfect. I think there's quite a lot of configuration that we need to do to turn this into a better representation of the data. But I personally think the pheatmap starting point is better. I prefer not scaling the data by row if at all possible, although I like having the option to do it if I want to. Okay, so what we're going to do is we're going to play with this now for the next 20 minutes or so, just to try and improve it, just to try and get it looking like a kind of nicer figure. So we're going to change the row names to be human readable gene names, and we're going to change the size of them so that they're actually readable, legible. And we're going to mess with the color scheme a bit. We might investigate whether we can scale by row. I don't know how easy it is to do with pheatmap. I can't remember. But in order to be able to do this, we need to read the documentation for pheatmap. So let's look at the help for the function. So here we go, a function for drawing clustered heat maps. And you will see, here we go, that pheatmap has an awful lot of arguments. It's very, very configurable. We can change lots of things about it. Let's kind of set about that. Let's begin to think about how we're going to change these things. So the first thing I think that I want to do, and this isn't actually something which we're going to change in pheatmap, is I want to change the row names. And in order to do that, the easiest way to do that is to change the row names on our matrix, to be honest. So if you look at the data, the data that we've pulled out in the same order as it appeared in Anno DF3. So we can change the row names of our matrix. Let's go back to our console for a second and have the start of the matrix, data for HM. So you'll see that effectively these are in numerical order because they're in the same order as they were originally in our Anno DF3. In order to change those row names, we just reassign them. This sounds really easy, but we are relying on the fact here that data for HM. What we're relying on the fact that the rows in data for HM are in the same order as the rows in Anno DF3, which we know that they are because we haven't rearranged them. But it might pay us to be a little bit more careful about this. I'm just going to do it in this straightforward way right now because I don't want to get sidetracked too much. But that's actually is a slightly risky maneuver without being more careful about making sure that these gene names that I've made the row names here actually do correspond to the ensemble gene IDs that were there before. And if I was, I think I would probably be a bit more careful about this if I was using, well, I definitely would be a bit more careful about this if I was using this for anything meaningful. And so now if we go back to pheatmap, now we have on our row names, instead of having ensemble gene IDs, we now have our gene names, but they're still not very legible because they're very large. So they're much larger than they need to be, so they all overlap with each other. You could relieve that slightly by making this figure larger. So if I zoomed it and then made it the height of my screen, and you can't see all of my screen, so this is going to drag off the bottom of my screen, but if I make it the height of my screen, then those you can see the gene names start to pull apart as it gets more vertical space to work with. The gene names start to pull apart from one another and it starts to become a little bit more legible. But actually, I think what we want to do is make those labels smaller so we can read them. Maybe in this stretched out version rather than the tiny, tiny version in my RStudio window. But still, we need to resize them. So the help file will tell us how to do that. It's just a case of finding the right argument for pheatmap. And all of those arguments are all laid out here, so you can read through them and read a description of them all, and there's lots and lots and lots of them, because as I've already said, this is a very configurable package. So let's have a look for something which will change the font size of the row. La 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 la. There we go, font size row. Okay, font size for row names. So it defaults to the base font size for the plot, so the font size that is used all over the plot, which by default is 10. But we can change it to be its own value by setting this argument for pheatmap. So let's change font size row, and let's set it to, let's make it quite small, let's set it to 4 and see what difference that makes. Okay, so we'll run that. And there we go, that's resized that font size. They're still a little bit cluttered, but again, if I use my full screen real estate now, you can see that they now no longer overlap one another, and they are legible. I can see those gene names and I can read them on my screen, so that's quite that's okay. That's quite a nice way of setting this up. That's got us those the row names now are the actual gene symbols, so they're a bit more human readable, and we've made them smaller so that they're legible. Okay, so to me, the next thing to do now, there's a couple of things I want to do. The first thing is just to see if we can go back to scaling by row just as an option. Yeah, so scale. So see, the scale argument is a character indicating if the value should be centered and scaled in either the row direction or the column direction or none. By default, this is none, whereas by default in the base heat map, it's scaled by row. So we can turn that on if we want to by adding scale equals row to, that's a character, sorry, so it should be in quotes. So by adding scale equals row to our call to pheatmap, and this will then scale on the row rather than leaving it unscaled. So this recovers that very kind of dichotomous view of gene expression where 3 of our samples are going to have high expression and 3 of our samples are going to have low expression. And you can see now that this centers the data for each row around 0. So you've got samples which have expression higher than the mean and samples which have expression lower than the mean, effectively, in each row. And generally speaking, you can see that you've got 3 yellowy orange samples on one side of the heat map and 3 kind of yellowy blue samples on the other side of the heat map. So this is showing you, okay, well, here's a set of genes which are have higher expression in these 3 samples, which, looking at the numbers, these are the mocks. So you have higher expression in the mocks in this set of genes at the top, and then in this set of genes at the bottom, we have higher expression in the infected, in the SARS-CoV-2 infected cells. So that's quite nice. I don't mind this color scale actually for this, because the Z-score, the row Z-score, is a bit more of a diverging scale than just plain raw expression. This way of showing this data illustrates the fact that this expression here is changing between conditions a little bit more elegantly than the raw expression data does, but it's not the be all and the end all. So I am going to play with the color scale, although this default color scale is quite nice actually. I think it's worth thinking about colors a little bit at this stage. We've tinkered with colors in ggplot so far, although really only by using color names rather than using any kind of concept of a colour scheme. So I just wanted to take this opportunity, especially as I've kind of got 5-10 minutes of talking about colours more generally. There are lots of ways of defining colour schemes in R. There are lots of different ways available to you for doing it. One of the simplest and a way of getting access to nice color schemes is a tool called Color Brewer. So Color Brewer, there's a website for Color Brewer actually, colorbrewer2.org, which looks like this, which shows you the idea of the color schemes. So these are color schemes. They're originally designed for kind of segments on maps, which is what the examples are showing you in the website. And what you've got here is a set of nice, or relatively nice, color schemes based on different kind of different properties. So you've got these are sequential color schemes. And they're called single hue or multi-hue. So single hue is working through just a single color palette, whereas multi-hue, you've got different colors at the different extremes. But these are sequential, so there's some progression, and there's not a point of divergence in these color schemes. On the other hand, you can have diverging color schemes as well. So here's a set of sets of diverging color scales. So purple to red and blue to red and various other things. And then qualitative gives you these kind of panels of non-clashing colours that can be useful for things like categorization, so categories of data. So the Color Brewer palettes are quite nice. And there's quite a lot of variety available to you by using the Color Brewer palettes. And they are available in R in a package, as so many other things are. So in R, we are going to install another package. This one is called R Color Brewer. Colors spelt the American way with no U, and the R, the C, and the B of R Color Brewer are all capitalized. Sorry, Biop Manager ::: install to install R Color Brewer. I'm going to update none again. Let's load our package again. Back in at the top, library R Color Brewer to load it, and then the way that you use this then is to use the palettes. So we can display Brewer all. This shows you all of the available color palettes. So here are all of those. These are actually the palettes that you could see on the Color Brewer website, just printed out in R with the names that they have in R as well. So at the top here, you have the sequential palettes. And then here in the middle, you have the qualitative palettes. And then at the bottom, you have the diverging palettes. And so these are all of the color palettes that are available to you. To make an appropriate color scheme to use in R, I'm going to have to remember how, and I can't. So R Color Brewer. Let's have a look at the package. Here we are. So to get the colors, you do brewer.pal and then pick one. And you pick one by the name. We need a number of colors. So the number of different colors that we want from that palette, and then the name. So let's have a look at the different ones that are available and pick one. Somebody said that they'd like to see it in black and white. So let's do greys. Okay, so greys with a capital G and spelt with an E. So let's pick that. So we want, how many colours? 1, 2, 3, 4, 5, 6, 7, 8, 9. There's 9 in the palette, and we want greys. So these are the hex values. So this is the way that you define colours in lots and lots of programming languages and on the web as well. So if you look at the R Color Brewer website, you'll see that for these colours, we're seeing the hex values for these colours represented on the website as well. So this is the way that you need to define your colours. So you don't very often define colours by name, you define them by these hex values. So this is a set of 9 greys, but for our heat map, so if we go back to our heat map, what you'll see is that this colour scale here is using a lot more than 9 colours. It's using loads more than 9 different values. The way that is defined is actually set out in the brewer.pal. Oh, you use color ramp palette. So look, they do it here. So this is the default. So here we go. This is the default color palette for pheatmap, and this is the way that it's defined. So what they do is they actually do what we're doing, what I've just done in my console here. So brewer.pal, they've done the same thing here, brewer.pal, to get the colors, but they've got it from the red, yellow, blue color scheme. So again, if we go back to here, maximize this, you'll see red, yellow, blue is down here at the bottom, red, yellow, blue. And they only take 7. There are 11 colors in that. So they only take 7, which gets you the middle 7. So from this kind of orange here through to this blue. And that's how they form their color scheme. But instead of going red to blue, they reverse it and they go blue to red. So that's what the rev is doing. That's reversing those 7 colors. And then this function, color ramp palette, that takes that set of 7 colors and extrapolates it into a much larger palette of colors. So instead of just having 7 colors, what you now have is, so this color ramp palette function creates a function that you then call with the number of colors that you want. So it's slightly complicated, this, but so they take 7 of those red, yellow, blue colors and extrapolate it out to a panel of 100 colors, which is then used to generate the heat map. So we're going to do the same thing, but we're going to do it with greys instead of with red, yellow, blue. So we'll replace the brewer pal argument in here with our own brewer pal argument, like that. We're going to get rid of the rev. I don't want to reverse this at this point. And I'm going to keep 100 colours. So what this will do is give me 100 greys rather than just 9. So this will give me 100 greys ranging from white at one end through to black at the other end. So what this means is that the low expression values will be white and the high expression values will be black. And I'll leave it that way round for now. And this gives me 100 colours. So I'm going to set this as a variable. So I'm going to call this blacks. And let's call it greys. Let's stick with that. And then I'm going to rerun my pheatmap. But we need to give it the colour argument. And that is just colour. Color equals greys. So the color palette that we just created on this line. Okay, and so that will replot our heat map, but this time in black and white, as requested. 100 Shades of Grey, well done. Very good. Yeah, that wins the comments for today. Okay, so there you go. That's our heat map in 100 Shades of Grey. But the Color Brewer palettes allow us to quite easily configure these heat maps with different color schemes. So if you wanted to use Peter's color scheme of choice, which makes no sense in this, but I'm going to do it anyway. I'm going to pander to Peter for some reason. So Peter likes paired. I don't know, well, for some reason, for everything, but it doesn't work for sequential data. But we're going to do it anyway because let's do some madness, right? Okay, color schemes always brings out the craziness in me, right? So rather than 9 greys, we're going to get 12 from paired, and then extrapolate that into 100 colors. This makes no sense. And then we're going to replot the heat map, but with pairs, just to show you that qualitative color schemes with quantitative data make no sense whatsoever. There you go. That's insanity. Look at that. Excellent. Weirdly, that's not hugely different from the kind of spectral color schemes that things like MATLAB use by default. That's a similarly insane color scheme. So yeah, that's crazy. Let's undo that, shall we? Let's do one final one just to settle on something that's not completely insane. Let's stick with sequential. Let's do purple, red, and let's go from let's get 7 of those. Right, okay, so final one. So our last scheme for today. So it's going to be, again, color ramp palette. We're going to get from the PURD, and we're going to get 7 of those colors. And then we're going to replot our heat map, and our color is going to be that. Okay. Oh, that's even See, that goes to show I have no design sense whatsoever. That's hideous. Oh, that's awful. Okay, you should take these decisions out of my hands and not let me design color schemes ever, because that's hideous. Okay, I'm going to stop there. Discretion is the better part of valor. I'm going to stop making up horrible color schemes, and I'm going to leave it there. Okay, so that is taking our expression data for our differentially expressed genes and producing a heat map with that data. And actually, I am going to do one last thing because I quite like it as a way of doing it. And I'm just going to fix that. So I'm going to very quickly fix this color scheme. And let's try and do it a bit better. Let's go blues, and let's not go purple blue, but let's go blue, just blues. Yeah, okay. So change that for blues. Okay, that's a little bit better. Still don't particularly like it, but it's a little bit better. And the last thing I want to do actually is show you that pheatmap's quite nice, and you can start to chop the heat map up into bits by the clustering, and it's relatively straightforward to do. So if we look at the help, for pheatmap and have a look down, we will find, I'm pretty sure, there we go. So we can use the tree, so use our hierarchical clustering dendrogram, which is what we have down the side and up the top, to split our heat map up into major sections by a number of clusters. So you can split it into a number of clusters by row and a number of clusters by column as well. And this is quite a nice thing to do where you have really obvious groups in your data. So we can cut our columns up into two groups using this cut tree cols argument. And that will put a little divider between your two groups of samples. So in this case, we know that these two groups are, we've got our mock infected samples on the right and our really infected samples on the left. So we can divide them up and kind of separate out our heat map a little bit by that. And we can also do it with, and here the clustering on the rows is also seems to be quite nicely dividing our genes into upregulated genes and downregulated genes. So we could do the same thing with the rows. We can cut the tree on the rows into two as well, and that should split us nicely into upregulated genes and downregulated genes, which it does seem to have done quite nicely. So that's the kind, the pheatmap package is kind of quite nice for this really quite quick and, relatively speaking, simple configuration of this kind of data, of these kind of visualizations. So I quite like pheatmap for this. When things start to get really complicated, when you start to want to add annotation in and things, which we could look at, but we don't have time to do today, then other packages are better. Things like complex heatmap begin to be better for those really, really complicated visualizations. But for these fairly straightforward things with relatively straightforward data, pheatmap is a pretty good choice, actually. Okay, and that really is where I'm going to leave it. So I'm going to sign off. Tomorrow, we will look at doing some functional analysis of these genes. So thinking about looking at gene ontology, enriched gene ontology categories and things like that. So that come back tomorrow for some functional analysis and some thinking about pathway analysis. Okay, I'll see you then. Signing off. Stay safe, keep well. See you tomorrow. Bye.